Hello, everyone. It's a great honor to speak at this conference. My name is Chen. I'm a co-founder and the CEO of RootPath. We are an early stage company with about 30 employees between our Boston headquarters and our R&D center in China. We started in 2017 and have raised about 20 million so far. Our core platform, which we call synthetic immunology, is a suite of technologies that allow us to read, write, and mine the immune repertoire, in particular, the TCR repertoire. We aspire to build this technology into a personalized TCR-T therapy for solid tumor, and we consider that our lead program. But there are also other opportunities in other disease areas with other ways to use our platform that we may explore in the future. Our main motivation to develop such a synthetic immunology platform and the personalized TCR-T therapy comes from the success and the limitation of TIL therapy, where tumor-infiltrating T cells are isolated from a fresh tumor sample, expanded ex vivo, and reinfused back to the patient. It has proven efficacious in highly immunogenic tumors such as melanoma and cervical cancer, which shows that polyclonal T cells programmed by patient-derived TCRs, some of which are tumor reactive, can exert clinically meaningful anti-tumor effect. And because these TCRs are patient-derived, they are also very tolerable and very safe. But the limitation of conventional TIL therapy is that in moderately immunogenic tumors, which are the vast majority of tumors, it has not shown meaningful efficacy. Presumably because in these tumors, tumor-reactive T cells within the TIL population are really rare, probably in the range of 1 in 100 to even 1 in 1,000, according to some reports. And since these tumor-reactive T cells are more likely to be highly exhausted, they tend to expand less well and get further diluted during the expansion process. So the question is, how can we identify those rare tumor-reactive T cells or their TCRs, even though those T cells themselves are highly dysfunctional and basically barely alive? Well, with the advent of single-cell sequencing, it is now theoretically possible to obtain paired TCR sequences of every TIL cell, including the highly exhausted ones, and thus digitizing the TCR sequences along with the transcriptomic information of the cells. And if we can somehow identify which TCRs are tumor reactive, we can then make TCR T cells using those TCRs and create billions of anti-tumor T cells for infusion. Like TILs, these TCR T cells will be polyclonal because we will use multiple TCRs to engineer the peripheral T cells, and which makes it very hard for the tumor to escape. Now also, like TILs, these TCR sequences are derived from patients' own TCR repertoire, eliminating many safety issues and are expected to be highly tolerable and safe. But unlike TILs, within which population the fraction of tumor-reactive T cells, as we discussed, is small and uncontrollable. Here, we can effectively ensure every single T cell we put back to the patient's body is tumor-reactive. And of course, this sounds a little bit like fantasy, but allow me to walk you through this process and pinpoint the core challenge and show you how our unique technology can address it and turn the scheme into a reality. So let's start with reading the full-length paired TCR sequences. This can be done by several commercially available platforms now, such as the 10x genomics system. Essentially, for each T cell, we can sequence and count every mRNA transcript. And these transcripts usually include the TCR alpha chain and the beta chain, and that gives us the TCR sequence, but also include other transcripts that are more indicative of the T cell's phenotype, such as activation markers and exhaustion markers. And with a typical 10x experiment, we can analyze 1,000 to 10,000 T cells this way at a total cost of a few $1,000, $2,000 to $3,000. So this is quite robust and affordable. But the problem is that we cannot tell which TCR is tumor reactive or neoantigen reactive. The TCR sequences themselves are by and large meaningless text strings to our human eyes. And the transcriptome information is a little bit helpful, but far from sufficient. So reading the TCR sequences is only the beginning, it's not the end. To know if a TCR is truly tumor reactive or neoantigen reactive, we have to synthesize it, put it in the T cell, and actually do the experiment. The problem is synthesizing each TCR, even using the smartest method available today, still costs about $100. That itself doesn't sound much, but if you consider we have to synthesize thousands of TCRs, which is necessary for the discovery of the rare tumor reactive ones, the costs quickly accumulate to north of $100,000. And that is obviously too much as part of a personalized therapy. So roughly speaking, our challenge is to reduce this cost by at least 100-fold. And this is the core challenge I was alluding to a few slides ago. And so how do we solve that? We turn to microarray-based DNA synthesis, exemplified on the right, which is available from multiple vendors, where we can custom order tens of thousands of DNA fragments at a time. And the per fragment cost there is indeed 100 to 1,000-fold cheaper than conventional synthesis. But still, there are two key problems we have to solve before we can make use of it in TCR synthesis, high throughput TCR synthesis. First, like conventional DNA synthesis, the fragments are only 200 to 300 bases long, and the variable region of a TCR is about 1,000 bases long. So we have to assemble 4 different fragments together to make the full-length TCR gene. And that brings up the second problem, which is all the DNA fragments in those syntheses come in a big mixture. So when you do gene assembly, all the fragments for all TCRs are in one test tube. So you have to somehow give instructions to these thousands of molecules so that only the 4 fragments belonging to the same TCR get ligated together in the right order, as shown at the bottom of the right panel. And this is extremely challenging. And if you're not meticulous with the sequence design and experimental procedure, you get all sorts of incorrectly ligated TCR, which is schematically shown in the mosaic to the right of the panel. So let's take one step of this process as an example to briefly illustrate this challenge. As we said, we break each TCR into 4 fragments and we assemble them together. Here, let's just focus on the ligation of the first fragment to the second fragment. In this illustration, different TCRs are shown in different colors. For example, the first TCR is showing green, the second TCR is showing orange. And what we want is green to be ligated to green and orange to be ligated to orange, and so on and so forth. And the way we ligate DNA sequences molecules together is by creating so-called sticky ends. To ensure correct assembly, we have to make sure every TCR have a different sticky ends, and they have to be different enough. However, this is tremendously challenging because in many cases, we have to design thousands of these sticky ends and make sure they do not cross-hybridize. And to make things a lot harder, in many cases, the sticky ends actually have to encode amino acid sequence. And different TCRs may have very similar or exactly the same amino acid sequence in this region. So there is very little room to create sequence diversity at a DNA level. And the sticky ends for different TCRs are inevitably very similar to each other. As a result, although the intended ligation can happen, shown in the thick black arrows, unintended ligations, shown in the thin gray arrows, can also happen. So you can imagine when thousands of pairs of these fragments are in one mixture, how hard it is to avoid the incorrect ligation. What we were able to do, without going into technical details, is first develop a biophysics-based, experimentally tuned algorithm to predict the strengths of DNA hybridization between 2 nearly complementary sticky ends with exquisitely high accuracy. And through an efficient optimization program, we were able to find a few thousand sticky ends like this that encode the right amino acid sequences, but also support precise orthogonal ligation. And lastly, we developed a proprietary experimental process to ensure the accurate assembly, to the point that 10,000 TCRs can be simultaneously synthesized by 2 to 3 technicians in a week. So to date, we have synthesized many TCR libraries like this, and we have rigorously characterized the synthesis product by next-gen sequencing. Of course, we measure many metrics, but the most important ones are cost, accuracy, and uniformity. We measure cost by how many TCRs you can synthesize with a $1,000 budget, which we think is a reasonable budget for a personalized therapy, for part of a personalized therapy. And using existing methods, including the very smart methods published to date, you're only getting a dozen each TCRs. But with our technology, we can easily get more than 1,000. And without going into much details, it's fair to say that the accuracy and the uniformity of the libraries we synthesize are more than good enough. And once we synthesize these TCR libraries, we can put them in expression vectors such as lentiviral vectors and transduce them into a T cell line or a population of peripheral T cells from a donor. In a sense, those initially dysfunctional TILs are now reborn into the flesh of vibrant, highly functional T cells through the synthetic process. And these reborn or synthetic TILs can then be co-cultured with tumor cells or tumor surrogate cells as target cells. And within as short as 24 hours, if a TCR recognizes any antigen presented by the target cell, the synthetic TIL that carries this TCR will express a reporter protein, such as a fluorescent protein. And these cells can be sorted out by FACS, and a standard next-gen sequencing of the TCRs in those sorted cells will tell us which TCRs are truly tumor reactive. In short, our synthetic immunology platform enables us to not only synthesize thousands of TCRs at a time, but also functionally screen thousands of TCRs at a time. And with these technologies developed, it's now clear to see how we can turn this personalized TCR-T therapy into reality. So basically, through single-cell sequencing, we can acquire the TCR sequences of all TILs and then synthesize those TCRs, put them in reporter cells. On the other hand, we can prepare tumor cells or tumor surrogates, such as autologous antigen-presenting cell pulsed with tumor antigens, neoantigens, for example. And then we can co-culture these cells and identify the TCRs that are tumor reactive or neoantigen reactive. And then we can use 3 to 5 TCRs from this process to make personalized polyclonal, or we can say oligoclonal TCR-T product. There are different flavors of this approach depending on where you get the T cells, from TIL or from periphery, and how do we prepare the tumor cells or tumor surrogate as a target. And each flavor is suitable for a different set of indications and clinical scenarios. And these flavors basically make up our pipeline. And we have identified a few indications, such as colorectal cancer, that are particularly suitable for these therapies in terms of the underlying biology, the manufacturing pipeline, and the feasibility of good trial design. And right now, we have completed technology development, and we're in the process of preclinical proof of concept and optimization of the manufacturing process. We expect to enter clinic next year through investigator-initiated trials in China, which is a regulatory green channel that allows us to get first-in-human data quickly with moderate capital investment. And in parallel, we will complete IND-enabling studies in the US over the next year and year and a half. The plan is that by the time we obtain IND in the US, we will already have sufficient human safety and efficacy data, which will allow us to greatly accelerate further clinical development. And compared to other personalized immunotherapies and cell therapies for solid tumors that are being tested right now, our approach offers orders of magnitude advantage in terms of the number of TCRs and the number of antigens we can screen. And lastly, although personalized TCR-T therapy for solid tumor is our main focus, there are many other ways to utilize our technology that we would be happy to explore potentially with partners. As a summary, we believe TIL-based therapy can be highly effective for solid tumors if and only if we can identify tumor-reactive TCRs for every patient, and that our synthetic immunology platform is the best in class and perhaps the only practical approach for this purpose. And furthermore, this platform unlocks the potential of TCR-T therapy and builds the foundation of a powerful and versatile discovery engine for a variety of diseases. So Please feel free to reach out if you see any opportunities to collaborate. We are always interested in amplifying the impact of our platform through such collaborations. That is all I have to say today. Thank you very much for your attention.